Bioinformatics.
Okay, in this lab follow along, we're going to be doing the first part of the RNAseq differential expression analysis. And so you should have already gone through the process of installing the DESeq package, which may have been easy or a little bit more difficult based on what operating system you're on. Unlike some of my shell follow alongs, I'm not going to be hand typing everything. I'm going to show you how typical R scripts tend to be used and executed from a script window, just to kind of help you think about best practices for R and to help expedite the lab. So as I follow along in the lab here at the bottom, I will be executing the commands through the script window up here, and you will see the outputs in kind of the console or R terminal, if you want to call it that. So first things first, we're going to need to load the library DESeq. So unlike shell, where every time you install something, it's now in your path and available for use at any point, in R, every time you make a new script or you open R, you need to explicitly tell it, hey, load this package. Even though I've already installed it, load it so that I have access to those commands, because there could be overlap between commands between different packages. So if you're on Mac, the way you execute lines within your script are you go to the line. You don't have to highlight the whole thing if you just want to do one line. Just be on this line and hit Command Return. And you can see it sent it down to the bottom, and we're getting some output. For Windows, I believe that command is Control R, but you can always go up to the top and click over under Code and look at what is the hotkey for running a section here. So you can go ahead and look at where that is for you. Okay, so hopefully you downloaded the RNAseq folder. I have mine downloaded to the desktop. So what we need to do is we need to navigate and set that as our working directory. So this is similar to CDing when you're in the shell. So the command is set wd, and then within quotes, you can navigate to what the path is. So for me, it's, since I'm on a Mac operating system, my home directory /desktop/RNAseq. And if you're using RStudio, you can still tab complete. So that's nice and helpful. So we're going to go ahead and execute that. And notice it comes down to the next line after I execute it. So I'm just kind of jumping through these commands as we go. So we're in the right folder now. And what we want to do is we want to take this file that's within this folder called cat-mouse-count-table.txt. Now what this is the output of kind of what you would normally do on your server, where you would generate mapped reads to make a SAM or BAM file for every single one of your treatments. So you would sequence the RNA of different samples, you would map those RNA back to the mouse genome or whatever genome you're interested in, and then you would extract read information from your SAM-BAM file using a variety of different software. And what you end up with is a table where each column is one of your samples, each row is a different gene annotated from your genome, and the numbers within each cell for every sample, for every gene, are the number of reads mapping to that region. And so if you think about how mapping reads works, if you have transcripts that are in greater abundance when you sequence RNA, you're going to have, by chance, more reads of that transcript relative to lower transcribed genes. And so we use the number of mapping reads as a proxy for transcript abundance, which is another way of saying the amount that a gene is being expressed. And so that's what this table is. We're going to read that table in using read delimited. So this is a way of taking a table file and converting it to an R data frame. And so it's within the folder data, cat-mouse-count-table.txt. It's a tab separated file. And we're going to put strings as factors, which is true. This is to kind of help avoid issues with gene names being treated kind of funny. And then we also are going to say that we have row names in the first row. So we'll go ahead and execute that. And just like we do in shell, let's head the data and see if what we actually get makes sense to us. And so here we headed the count data that we just made. Again, if you're new to R, you can see this little arrow is saying whatever the output of this function is being sent to this count data word that I just made and set as basically a variable to contain the contents. And so if we head the contents of that variable, we can see here are a bunch of gene names. This is actually 36,000 rows. And here are our 4 treatments. So there were 842 reads mapping to this gene for this individual here, while only 437 mapped for this individual. So these individuals, what are they and what do they mean? Well, these are just the sequence IDs. We didn't make the names super fancy when I made this data set for you. But if we go and look at the file within the RNAseq data folder, there is a cat's mouse phenotype file, and it looks something like this. And what this is metadata. Now, metadata are basically a lot of your information on experimental design, how each sample was treated. It's not going to be involved directly in the analysis, but you will pull information from these things often for many types of analyses. Specifically, what we're interested in here, and you could open this up in Excel if you wanted a cleaner table, is the knockdown status. Because, again, if you read the beginning of this lab, there are mice that had a knockdown for this splicing factor and mice that didn't. And so these sequences here are the ones that have this shRNA knockout. And then we have the control that's not a knockout, basically. And so we can look at the column names here. So this first one is a knockout. The second one is a control. Third one is a knockout. Fourth one is a control. And so what we want to do is we want to make a little miniature table to pass that information to our DESeq analysis. And so we're going to do this really simple. We're going to make a variable called group, and we're going to use this function to concatenate several different words together into almost like a one-line table. And it's going to correlate with the order of our columns. So the first column is a knockout, second is control, third is a knockout, fourth is control. And so we will execute that. Now we have all the info that DESeq wants to make basically a DESeq data set. And they call it a new count data set. And what that involves is the first input is your actual raw count data, so this table of your reads. And the second is group, or whatever you named it, which is basically information in a simple row of what each column belongs to as far as treatment is concerned, whether it's a knockout or a control, whether it's a male or a female, whether it's a inbred versus outbred mouse, which will be something we work with later. So when we execute this, we now have a variable called DDS. Now, DDS is just the name we've given to that new count data set. And so we could execute DDS and see what it is. And we're going to get kind of just a brief kind of synopsis at the top of it because it's a little more complicated than just a table. It's got multiple layers to it. And we'll talk more about what these things are. But you can see the samples that are in here. We haven't identified any size factors, anything like that yet, which is what we're going to do now. We're going to have this string of things that look kind of funny where we do something to the DDS and save it over itself, basically. This is something we typically don't do in Shell, but in R it's quite common to do this. So there are all these different things like size factors and dispersion rates that we're going to calculate from the data contained in the DDS data frame. And then we're just going to save over that DDS data frame with that new calculation added into it. So if we go ahead and execute this, we have now estimated size factors for DDS. Now, we can use the function size factor to look specifically at those within the DDS frame. So we'll execute that. And here we can see that of our 4 treatments, we have size factors ranging from 1.7 to 0.6. Recall from lecture that when you sequence things, not all things are sequenced equally. Just by chance, one of your libraries or samples might get sequenced more than another. It would not be fair to look at just the raw number of mapped reads without taking into account the total amount of reads that were sequenced for each individual. If one was sequenced 1,000,000 times and one was sequenced 100,000, you would expect there to be a 10 times difference in just the number of reads mapping, even if there was no expression level difference. And so what we're doing is we're estimating the size differences of the total samples so that we can then use that to normalize so that they're on a level playing field when we compare the estimated expression level. So I've got some questions here for you to think about. What's a simple way to correct for a library size difference? In kind of a really pen and paper strategy, you could take the total number of reads per sample and divide every single number of mapped reads for each gene by that large number. And that would be one way to kind of normalize by the total library size. Here we're generating a factor based on the difference from the mean, where the larger the library, the larger the factor. So you could almost think of all the reads being divided by this number as well. So if you start with more, you divide by a bigger number. If you started with less, you divide by a smaller number. And so that's kind of the nuts and bolts of that. So we could also, again, just to show you that there's different ways to kind of get at this information, if you didn't believe me that these are the larger ones, we could actually use the R function sum to just sum all the read counts for one of our columns, for one of our individuals, to get kind of an estimated library size. So here, this is for individual 33, this first one, with the size factor of 1.2. So if we executed that, there are, again, this is a small data set, 1.4 million reads. Typically, I like to see something more along the line of 50 to 60 million reads per sample. And so let's do one of the ones that is on the smaller size. This one only has 700,000. And so, again, if you were trying to remember, well, do the size factors, are they bigger for the bigger libraries or is it inverse? You could just check it and go, okay, well, this one, clearly, the bigger the library, the bigger the size factor. Okay, so as we scroll down here, the next thing we need to do is estimate dispersion. And so dispersion can be kind of a tricky one initially. And so what dispersion is a plot of the mean expression level for every single gene versus the variance across the data set for that gene. And so if we don't take into account which treatment things belong to, and you can read more about this, and do something really simple where we're just blind, we're just going to calculate the dispersion for our whole data set without thinking about individuals and treatments, we can estimate it and plot this out to take a look at it using plot disperse estimates. So this will help me kind of explain this. So on our x-axis, we have mean normalized counts. Now, this is the number of mean reads basically mapping to any given gene. Every single gene is represented here as a dot, so there should be around 36,000 dots contained within here. So lowly expressed genes are on the left, highly expressed genes are on the right. Dispersion is basically a way of summarizing the variance. And so the higher up these dots are, the more variance there is in the expression level within the data set for that gene. The trend that we see is that the more lowly expressed genes, the ones on the left side of this plot, tend to have much higher variance, making it harder to make statistical determinations about whether they're differentially expressed or not. Also, if you go back and kind of read more about the lab, you'll see that not only do these lowly expressed tend to be more highly dispersed, it's actually kind of got a reasonable pattern to it that is due to what we call shot noise. And so for this reason, unlike most biology, we don't tend to use a lot of technical replicates because it doesn't really help us overcome that shot noise at the lower end of our expression level. So we tend to mostly just focus on upping bioreps when we think about RNAseq. So that's if we just treat the whole data set as a whole. Typically what you want to do is you want to estimate your dispersion for each condition individually. Again, we only have 2 reps for each condition, so this is less than ideal. You normally like to see at least 4 or 5. But we can estimate that, and then we can plot them individually. So here is the dispersion for the control group, and here's the dispersion for the knockout group. Same general trend, but this will help us when we calculate our statistics to have a little more precise information about dispersion rates. So now that we have the dispersion calculated and the size factors, we can actually run the basic statistical test here, which is a negative binomial test. That helps account for this kind of predictable dispersion at the lower end of expression level. You don't need to get into that though. What this is saying is we're going to perform a negative binomial test, which is going to be testing every single gene individually between the control and the knockout groups to see if there's a significant difference in the expression level or the number of normalized mapped reads. And so this can take a second on bigger data sets. It should be pretty quick on this one though. And so it's done. So let's take a look at what we saved as our results file from this and look at a summary. So summary gives you a lot of just basic descriptive statistics on any table. This is going to look pretty funky, just to warn you. So we've got a summary for all the different columns in our results, which include the ID, the base mean, so basically the mean expression level for a gene across the data set, the base mean for our A group, which is the control, the B group, which is knockout, the fold change between A and B, as well as the log 2 fold change, which is a nice way of summarizing it, p-value and p-adjusted. Now, I ask you to kind of look through here and see if anything looks funny in the stats for log fold change. You could look through here and be like, why is the log fold change minimum negative infinity? That seems weird. We've got NAs here. We've got maxes that are infinity. There's something kind of funny with our results data. And so let's head the results data, which shouldn't be in any particular order, and take a look at it. And right here, this should jump out at you. We've got genes that have an expression of 0 across the board. So that means the log fold change is NA, the log 2 fold change is NA, there's no p-value. This is going to kind of make our data set a little bit messy. We've tested things that don't really exist as far as our samples are concerned. So I ask you, why might some expression values be 0 in both treatments? Well, this is a mouse muscle tissue with knockouts. There are some genes that are just not expressed in mouse muscle tissue, right? So if it's a gene that's only expressed in the brain, you would expect it to be 0 in both treatments and not be useful to compare anyways. So there's going to be always a subset of genes when you're doing expression comparisons that are just going to be off because they're not active in the tissue or developmental stage that you're looking at. So what we really want to do is we kind of want to remove these zeros, clean these things up, and then kind of adjust our test. So what we're going to do next is we're going to take a better look at what we have by ordering everything by our adjusted p-value. The reason we have a p-value and an adjusted p-value is because we've just tested 36,000 rows, and so false discovery rate from multiple testing is a real problem. And so we have to adjust our p-value by the number of tests and things like that we're doing. So here you can see in a vacuum, the p-value for this gene was 0.03. But after we adjust it to correct for false discovery rate, it's actually 0.5. So we are much more stringent than we normally would be because we're doing so many different tests. So we're going to order by the adjusted p-value by calling the results and then ordering based on the results column, the $ is column, p-adjusted. And so if we head that ordered results, now we can see that we do have some really small p-values for some of our genes. We've got things where the log fold change is negative 4.98 with an adjusted p-value that's got 34 zeros in it. So pretty good indication that we do have some differences somewhere in this data set. So the next thing I want to do is also count how many have an adjusted p-value less than 0.05. So nrow is a function that will count how many rows are in a data set. So what we're going to do is subset our data set and then count it, where again, going back to Boolean operators, the p-adjusted is less than 0.05 and is not NA, because we don't want to get the NAs in there as well. So if we execute that, we'll see that there are 201 rows or 201 genes where the p-value is less than 0.05, the p-adjusted value. How many do we have total? We could just do n rows on the results table without subsetting it. So 201 out of 36,000. So if you knew nothing about these mice, nothing about this knockout, rather than taking a dartboard and throwing darts at 36,000 possible targets to identify what genes are involved in response to this knockout or this knockout is functional for, you've now limited it down to just 201 potential candidates already. So that's one of the powers of these kind of large-scale comparisons. Okay, so everyone likes a little bit of nice graphics, and so we can actually plot our results as one of my favorite plots, which is called an MA plot, and it's plot MA, and we'll use the ordered version, and we'll set the y-axis with a minimum of -10 and a max of 10, and I'll show you why, and we'll go ahead and plot this. And so this is a cool plot. It shows quite a bit here. So the x-axis is the mean normalized count, same as the dispersion plot. So lowly expressed genes on the left, highly on the right. Each dot represents a gene. Log fold changes on the y-axis. So if there was a large change in one direction, meaning higher in the control or higher in the knockout, it's going to be represented here as being higher or lower log fold change. And you can see how that relates to just fold change as well. So things that have a significant difference, so where the p-adjusted value is less than 0.05, are highlighted red. So remember how I mentioned that the dispersion, particularly at lowly expressed genes, makes it hard to get statistical significance? That should be quite evident here. The farther out you go to the right, the more highly expressed genes, the smaller log fold change you're able to pick up as being significant. When you look over here at these lowly expressed genes, you really can't pick up anything. So something I like to do, because again, we're looking at potential false discovery rates and trying to correct for that and being really stringent, there is no chance that we will really ever get useful information out of genes where the number of mapped reads across the data set was averaging less than 5. It's almost non-existent. It's almost, you could consider it an artifact. So what I like to do is I like to go and remove all the zero count genes as well as the ones that are ridiculously lowly counted, that they're almost zero. So what we're going to do is we're going to take our results and we're going to subset it so that anything with a base mean expression level less than 5 is removed. In other words, we're going to keep anything greater than 5. And now if we look at how many we have, I used dim this time, which gives you the number of rows and number of columns, the dimensions of your data frame. We went from 36,000 to now 6,200 potential genes that are actually worth statistically testing. And so what we can do now is we can readjust our p-value based on the new size and new number of comparisons, because 30,000 of those weren't even really valid comparisons. And so we're going to use p-adjust of the p-value using this BH method. So if you wanted to look at the help menu for p-adjust and see what the BH method is, you could do help p-adjust if you were curious and read about it and see that the p-adjust is actually going to be using the Benjamini Hochberg, in parenthesis here, it's BH is the shorthand for that, and it's also known as false discovery rate. You could use one of these other ones as well. You could use home if you wanted to. So we're going to run this and readjust our p-values. And now we're going to, just like we did before, report out how many are not NA and have a p-adjusted value of less than 0.05. Remember, before it was 201. So we've added about 31 additional candidate genes that we think there's significant difference in expression within mouse tissue, muscle tissue, after knocking out that gene. So now that we have all of that, we want to save it. Those are our results, basically. We've got 231 genes that had a p-value less than 0.05, and we've got 36,000 that were either not worth comparing, or you could think about the 6,000 that were worth comparing that showed no significant difference, or we don't have evidence that there's a change. They're not affected by that knockout, meaning they're probably not related to that pathway. So what we're going to do is we're going to save some tables because we're going to use these things later. This is kind of just the first step. And so we're going to write a table, and we want to write the data that's contained within the results 5 after we order it by the p-value here. We're going to save it within the results folder, and we're going to name it DESeq mouse cats all genes. We're going to make it a comma separated. We don't want quotes around any of our gene names or anything, and we're not going to add in row names. So if we execute that, it will save it. You can go look at your folder right now, and it'll be there, and you can open it in Excel and explore it. But then I also like to make a table where we just have the genes that were significantly different. So here we're going to use that same code to subset out everything that's not less than 0.05. We're just going to keep things that are less than 0.05 and not NA, and we'll give it a different name, and we'll save that as well. And so that's where we're at right now. In the next lab, in the next lecture, we're going to talk about what to do with these tables, how to do some other kind of fun things with them to start to think about biological implications. So hopefully that was helpful, and just let me know if you need help with some of this.